back, students, to this chapter, and in this module we shall be having a look at plasmids. Now let us move ahead and study what plasmids are. Here you can see a bacterial cell. Now let us have a look at the inside of the bacterial cell. Here you can see the bacterial chromosomal DNA, and you can also see a circular DNA, which is nothing but the plasmid DNA. Now let us take a look at the qualities of this plasmid DNA. The plasmid DNA is small, it is extra chromosomal, and it is a circular form of DNA. So plasmids are small, extra chromosomal and circular DNA. They are also autonomously replicating, and they are not needed for survival. So plasmids are small, extra chromosomal circular DNA which are capable of autonomous replication, and they are not needed for survival. So these are the qualities of the plasmid DNA. So moving ahead, plasmids are occasionally found in bacteria and yeast, whereas plasmids are rarely found in plants and animals. So bacteria and yeast are the organisms where we can occasionally find plasmids, whereas they are found rarely in plants and animals. Now let us move ahead and take a look at this scientist known as Joshua Lederberg. So Joshua Lederberg was an American molecular biologist. Joshua Lederberg was the person who coined or introduced the term plasmid in the year 1952. So the credit of coining the term plasmid goes to Joshua Lederberg. Moving ahead, so here we shall have a look at a few more qualities of the plasmid. So plasmids show a size range of 1 to over 1,000 kilobase pairs. Next, they are transferable genetic elements. They are used for gene transfer processes, and they are called as replicons. Why? Because they are capable of autonomous replication within a suitable host cells. So plasmids are known as replicons. So here I have a bacterial cell, and here I have a plasmid in the bacterial cell. Now these plasmids generally carry genes for metabolic activity. So they usually carry genes that are related to metabolic activity of the organism, and they help for the survival and reproduction of the organism under unfavorable conditions. So these are the roles that are played by the plasmid in an organism. Now let us move ahead and take a look at the nomenclature of the plasmid, that is how the plasmids are named. It begins with a first letter, a small letter P, which stands for plasmid. Next, it will be followed by a few initials. Here we can see the initials as BR, which stand for the discoverers of this plasmid as Bolivar and Rodriguez. And then we have the number 322 that is designated to the plasmid, which is basically the 322nd plasmid that is being discovered by Bolivar and Rodriguez. Next, let us have a look at another type of nomenclature. Here I have a plasmid. We again begin with the small p, which stands for plasmid. Next, UC. Here these initials stand for the place of discovery. UC stands for University of California. And the number 18 stands for the 18th plasmid that was discovered at this place. So PUC 18. Here P stands for plasmid, UC stands for University of California, and 18 is the number designated to this plasmid. So let us have a look at the disadvantage of the plasmid. Plasmids, we have already studied that they are transferable genetic elements, but during transfer we need to insert certain DNA or certain genes of interest. Now plasmids are unable to accommodate long DNA fragments, so that is one disadvantage of the plasmids. So plasmids cannot accommodate long DNA fragments, and hence we constructed cosmids. Now cosmids are the plasmids that have lambda phage fragments with them. So cosmids are the plasmids having lambda phage vectors. So these are the new forms of cloning vectors that we shall be studying in detail in this chapter. So here we had a look at plasmids, and in the next module we shall be having a look at bacteriophages. So till then, take care, study well, and bye-bye.